And we're recording again just because James couldn't be here, so I'm gonna post these later I'll post them for everyone, but can't start the video. It's just strange, but okay, we'll go ahead and start in this. Okay, let's review a little bit of what we did just at the very end yesterday, last time we met. So we're talking about photosynthesis, and basically there were sorry about that. There are two reactions that we want to talk about. One was let me see if I can find the right slide. There are the light reactions, so that's what we covered last time. So you can think of it this way: in the word photosynthesis, this is the photo part of it, right? It involves light. And then the synthesis part is really the second part, the Kelvin reactions, and these are considered to be light independent. Now, in some textbooks, and actually in one of the diagrams, we call this a dark reaction because it can occur in the dark, but it doesn't have to be dark, right? So it occurs in the leaf, and it just doesn't need light. That's all it means. And we introduced this new thing, nicotine amide adenine dinucleotide phosphate, NADP. Now remember that this was one of these shuttles that helps us move electrons around. It was a lot like NAD, right? It could pick up 2 electrons and a hydrogen and become NADH. That's when we were talking about cellular respiration, or aerobic cellular respiration, I should say. And ATP is something we know about. So one of the things that comes out of the light reaction, does anyone recall what happens? What do we get out of the light reaction? What are the products essentially of the light reaction that are then fed into the dark reactions? Oh, yeah, ATP is one of them, right? So one thing we get out of it is ATP. Do you remember how, and now I know you have to rack your brains a little bit, how was ATP synthesized in the light reactions? I'm purposely not showing the diagram because I want you to think a little bit. Go back in your mind and think about how is that ATP made? No one? So it's similar to there's some similarities between the light reactions and aerobic respiration. What is can anyone think of any similarity?
They involve oxygen.
They do involve oxygen, right? So yeah, let's talk about oxygen for a second. That's a good. So in aerobic respiration, what's the role of oxygen? What does it do? It's what's called the term is the final electron acceptor, right? It's an electron acceptor because oxygen is so electronegative, right? So, and oxygen is found in the mitochondrial matrix. So it accepts electrons, and at the same time, each oxygen accepts 2 electrons and 2 hydrogens, and it becomes water, right? H2O, right? So you're generating water all the time in your cells when you're doing aerobic respiration. So the air you breathe in becomes water, right? It does not become CO2. Where does the okay, I know we're talking about, this is photosynthesis, but I'm talking about respiration, but just go back in your minds because it's a good review. Where's the oxygen and CO2 that you breathe out all the time? That's the waste that your cells produce. Where does that oxygen come from? Yeah, it comes from the glucose, right? And we've been using glucose over and over again, but of course, there are other things we can talk about. Your book, I think, briefly mentioned, fatty acids and amino acids can also be used as fuel, but to keep things simple, we're using glucose. So that oxygen comes from glucose because it's a carbohydrate, right? So a carbohydrate has a ratio of 1 carbon to 2 hydrogens to 1 oxygen. So it actually has quite a bit of oxygen. And so the oxygen in CO2 is all from the carbohydrate. Okay, so let's go back. We got a little bit off track, but that was a good thing. So oxygen was a final electron acceptor, right? We forget. And now it sits in the matrix of the mitochondria. And where does it get the electrons from? What's the thing that feeds electrons to it? We're talking about, again, we're talking about cellular metabolism, not right now, not the photosynthesis. There's a bunch of proteins that are found on the inner mitochondrial membrane. Yeah, they carry, what do they carry? Electrons. So it's called the electron something. Yeah, right. Electron transport, right? Chain. So electron transport chain, ETC. So in aerobic respiration, we have the electron transport chain. The electrons, remember, they move down in energy as they go to successively more electronegative molecules. And what do they do when they do that? As the electrons move down, remember we had this sort of picture. Your book had these pictures of electrons as being sort of like water. So I'll draw them on the board. So they sort of move downhill, right? So if you have an electron up here, it enters, of course, it's negative. It enters into the electron transport chain. It ends up here, and it goes to oxygen, right? It combines with oxygen, and then and two hydrogens that are in the matrix, and you get water. So what's the point of the electron transport chain? What's the point of that? What does it do? It must do something. You have all these proteins in there. They're in the inner mitochondrial membrane. Remember, the inner mitochondrial membrane has a really increased surface area. You guys remember that? You have the cristae, so it's a lot of infoldings. So it's very important. All your cells have it. What was the whole point of having these electrons move down the electron transport chain? Yeah, to make ATP. And how do you make ATP? Does anyone remember? Okay, here's. Go ahead.
ADP turns in energy.
Right. So you're going to take ADP, right? ADP plus inorganic phosphate, which is PO4, right? Inorganic simply means it's not combined with carbon, hydrogen, and so forth, right? And then that makes ATP. But what's the mechanism by which we make the ATP? So if we look at a mitochondria, it looks like this, right? The double membrane. And so this is the cytosol out here. This is the matrix. Does that ring a bell? That's the fluid in the middle. And what's this space here between the two membranes? Yeah, it's the intermembrane space. That's a perfectly good name, right? Inter means between, membrane, space. So the electron transport chain, where is it located? It's on the inner mitochondrial membrane, right? So everyone remembers that, right? So you got all these proteins all over this thing, right? So electrons, they come from NADH, right? It gives up the electrons. It becomes, oops, NAD. So it's giving up electrons. And we have FAD and FADH2, right, also. But so it's giving electrons into the electron transport chain. The electrons move downhill. Eventually they get to, in terms of their energy, they eventually arrive at oxygen, and we have water. But the point wasn't to make water. This is actually kind of a waste product in a sense, right? That's not the whole point of this, right? The point was to make ATP. So what are we harnessing from the electrons as they move downhill, downwards in terms of energy? We actually have to do something. We have to pump something, right? Okay, the analogy I gave in class was that if you have, what's that? Go ahead.
Hydrogen.
Yeah, you have to pump hydrogen ions, right? Exactly. So the analogy I gave is with some places, what they do is they have power plants to generate energy, right? Like a coal power plant or even solar or something like that, or wind turbines, and they'll use that energy. The energy doesn't always correspond to when people need to use energy, right? Like, for example, the power plants, like a coal burning plant might work all night. People don't use a lot of energy at night, right? So what do they do during the night? They'll pump water uphill into a reservoir, right? And then the water's held by a dam. And then when the energy is needed, they'll turn the turbines on and so forth. So what we're doing here is we're going to pump, it pumps hydrogen ions. You guys remember this now? Hydrogen ions into the intermembrane space. So what happens is you get a lot of hydrogen ions in the intermembrane space. And of course you have some, right, in the matrix. It's not like there's none. But if you look at the concentration, it's higher in the intermembrane space because they're constantly being pumped in there. And then what we have, we have a chemical gradient, right? Can everyone see that, right? Chemical gradient means you have a difference in concentration across the membrane. And we can harness the chemical membrane to do work for us. There's actually also going to be an electrical gradient because as these hydrogen ions are pumped up here, now there are negative ions, which we generally don't talk about, but they're in here. There's chloride ions, right? They're negative. There are proteins. Those generally have a net negative charge. So you also have an electrical gradient where it's positive up in this intermembrane space and negative inside here. That's also going to be very powerful because it's going to essentially, the hydrogen ions are repulsed by each other, but they're attracted to the negative ions in the middle, in the matrix, right? So there's a very powerful electrical field across this inner mitochondrial membrane. Okay, so the hydrogen ions will want to move downhill, right, into the matrix. So this is the intermembrane space. This is the matrix. And what do they move through, and how do we harness that energy? So here's the membrane. They actually pass through something that looks a little bit like this. This is obviously just, I can't really draw them. So there's a channel, right? And it goes through this thing. And then there's this rotor that turns around and around. And what does it do? Yeah, it's ATP synthase, right? Well, good thing we're doing this review. ATP synthase, right? So okay, does that ring a bell for anyone, like ATP synthase? Okay, so remember I showed a video like this, the hydrogen ions go through this, there's a rotor that goes, this thing goes around, this thing spins around and around. It causes the conformational changes in this protein down here, and then you have ADP. So someone mentioned this earlier, ADP, adenosine diphosphate and inorganic phosphate coming in, and what you get coming out is ATP, okay? All right, so why are we talking about this? Well, because I want to remind you that in the light reactions, there's an electron transport chain, okay? Remember that? Between the two photosystems, right? So there are two photosystems, photosystem 1 and photosystem 2. Which one was discovered first? Well, no. Photosystem, from a functional point of view, photosystem 2 comes first, right? But in terms of discovery, that's why, you know, historically photosystem 1 was found first, right? That's why it's called photosystem 1. But then they found photosystem 2, and they're like, uh-oh. Because what's the order? Photosystem 2 actually comes before photosystem 1, right? So you're correct there in terms of the way things move, right, throughout the system. Okay, so, all right, so what's going to happen? Remember, there's an electron transport chain between photosystem 1 and photosystem 2. And so the electron transport chain essentially does the same thing, right? It's going to pump hydrogen ions. And then we'll have ATP synthase, and we'll make ATP. So that's how we get the ATP from ADP. Now, we're also going to harvest electrons in the non-cyclic version of the light reaction, which is the only one we're going to discuss. The book briefly mentions cyclic light reactions. You're not responsible for that. We're just going to do the non-cyclic ones. So NADP becomes NADPH, right? And when it does that, it picks up 2 electrons. Does that make sense? Because if you add 1 electron, it goes from NADP as an ion with a charge of +1 to just NADP, and then you add a hydrogen plus 1 electron, right? So it's 2 electrons, right? Makes sense. So how do we get that? At what point do we get that second? Where do we get the NADPH? Is it directly after photosystem 2 or after photosystem 1, or where? Does anyone remember this? Do you have it in your notes, anyone? So we're going to harvest, we're going to make ATP. This is going to be super useful. This is going to be really, so remember the Calvin reaction is going to be a synthesis reaction, right? So we're going to need energy. So that, okay, that's great. We've got ATP. We're also going to need electrons. We're going to be reducing CO2, carbon dioxide in the atmosphere. Reducing means we're going to add electrons. Hydrogens will come with that too. So this is going to be our source of electrons and actually of the hydrogen also. So it's important that we make NADPH. The question then is, where is it made? Photosynthesis 1, right? Is that what you're saying? No. Yeah? Okay. Right. So right after so what happens is electrons move down in terms of energy, and then they get, so this is as they move from photosystem 2 to photosystem 1. Then the light hits the photosystem 1, right? There's a sort of an antenna with the pigment molecules, the chlorophyll and so forth. We have a reaction center, electron gets boosted again, and then it actually goes to NADP reductase, which is an enzyme. Reductase is an enzyme that's going to help with the reduction reaction, and then we're going to add the 2 electrons and 1 hydrogen to NADP, and it'll become NADPH. So basically our output from the light reactions will be NADPH and ATP. So we have our energy plus energy in the form of electrons plus a hydrogen, and those are going to go into our light independent reaction, which is the Calvin reaction. All right. Yeah. So just as a quick reminder too, so we understand so light reactions occur to the thylakoid membranes, right? These require solar energy and water. They produce oxygen. Because oxygen is split. Where does the oxygen come from? For the light reactions? I mean, yeah, I mean, what was it before it became, so they make oxygen, right? So plants make oxygen, right? But where do they make it from? What molecule was it in before? Water, yeah. So plants have a root system, right? So they drop water up to the leaf. All this is occurring in the leaf. But the water is actually, brought through the root system and the system of veins and so forth up to the leaves, right? So water is split to become oxygen. We make ATP and then NADPH. Okay, so we just talked about that. Now the Calvin cycle is going to occur in the stroma. Does anyone recall what the stroma is? No? Okay, so let me draw a picture. So if we look in a chloroplast, it actually has a double membrane. We have these thylakoids, right? And they form a stack. And remember these have a thylakoid membrane, and these thylakoids are actually connected to other thylakoids, right? The thylakoid space is the fluid within the thylakoids. Okay, so you look at these things, and each one of these little things is called a thylakoid. Let's see, my spelling's not that good. So thylakoid. Now, if you took a stack, what is a stack called? Does anyone recall what that is? Grana. Yeah. Well, okay. So grain Is singular and plural is graina. This is singular. And why is that? Well, singular. It's because they look like, to the anatomists that will look at this, they look like little grains or something like that, right? So graina means like a grain. Graina would be grains in Latin. Okay, so you have the thylakoid membrane. The thylakoid space is inside the thylakoid membrane, so it's a clear liquid there. The thylakoids themselves, they're going to have the pigments and so forth. So they're going to be greenish within the chloroplast because of chlorophyll. Now, where's the stroma? So we have a fluid out here, which is the stroma. Okay, so that's a clear fluid that's outside of the thylakoid space. So basically, we have a thylakoid space, the thylakoid membrane, and the stroma. So the Calvin reactions are going to occur within the stroma, right? So they're not, previously, everything we talked about was essentially within the thylakoid. We were talking about this, we were sort of zooming in on the thylakoid membrane, the thylakoid space, and the stroma. Now the Calvin reactions are occurring out here. What are they going to require? Well, it's going to require energy. Remember, it's a synthesis reaction, so this makes sense. We're going to have energy in the chemical energy in the form of ATP. It's going to require electrons and hydrogen and so forth. Why do we need the electrons? Because we're going to do a reduction reaction, right? CO2 is just from the atmosphere that comes in through this little stomata. Remember those little mouth-like things? That there's a pair of cells, pairs of cells on the bottom of each leaf of a plant. They can open up. They allow CO2 to come in. So reduction means we're going to add electrons to that carbon here, right? So what are we going to make? We're going to make a carbohydrate, right? So that's going to be a reduction of this carbon here in CO2. So we're producing through this reduction reaction carbohydrate. Note that it's just, instead of doing C6H12O6, which would be glucose, we're just giving the standard general formula, which is C1H2O1, right? That's the standard ratio for any sort of carbohydrate. 1 to 2 to 1. And then we're also, since we're going to be using up this ATP in a sense, we're going to be hydrolyzing this ATP. It's going to give up its energy. It's going to become ATP and inorganic phosphate. And then since this NADPH is giving up its 2 electrons, it's just a shuttle, right? Remember? It's moving these electrons around. It's going to become NADP, which is a positive ion with a charge of +1. Okay, so then here's our overview again. It's good to have overviews, right? We've got, this is actually the same thing as the picture I just drew on the board. We have water, which comes up from the roots. We have solar energy coming in. We have the light reactions occurring here with the thylakoid membranes. Water is being split, so we're getting oxygen. So that oxygen is useful for us to breathe, but it's also useful for the plant because they do aerobic respiration also, just like we do. They have mitochondria just like we do. Now we're getting out of the light reactions NADPH and ATP. So this is essentially harvested energy, right, from the sun. These go into the Calvin reactions, which is a light independent reaction. And then what's the point of this? Well, we're going to take CO2, which is found in the atmosphere, and then it becomes carbohydrate. We'll get the glucose, for example. And then the NADPH will become NADP, and ATP will become ADP, and organic phosphate. All right. All right, so then I just wanted to show you this. Again, we talked about this last time, but it's a very confusing diagram, but we can put it all together in this diagram. We looked at the general reaction. If we look at just the light reactions, remember light comes in. Here's the thylakoid membrane. Here's photosystem 2. Remember 2 comes first. So photosystem 2 it's been involved in splitting water. So we get oxygen, electrons, which is what we really want, and also this hydrogen ions that end up, when we remove the electrons, we also remove the hydrogen ions, right? So hydrogen ions are found now in this thylakoid space, right? So that's going to contribute actually to this hydrogen ion gradient, right? Okay, electrons are boosted in energy. They're fed into the electron transport chain. Now here we can see this element here is actually an ion, it's a proton pump, right? This element PQ, it's a protein. It's going to move hydrogen ions from the stroma, right, into the thylakoid space. Does that make sense? So we're building up, you see, we have two different things that are contributing to the hydrogen ions. The fact that we split off the H2O, and we took the electrons, well, that actually means we're donating hydrogen ions to this thylakoid space. We're also pumping hydrogen ions in here using the energy within the electrons as they move down in terms of their energy, potential energy. Here it goes to another thing. These are just proteins within the thylakoid membrane. Eventually they get to photosystem 2. Photosystem 2 also receives light. It has chlorophyll. There's a reaction center. We talked about this previously, right? Yeah, so the light you see is funneled towards this reaction center, which is a special chlorophyll, and the electron is boosted in energy. Right. And then this boosted electron goes to NADP reductase. So that's an enzyme. What's it going to do? It's going to reduce NADP. So it's donating, we're going to donate 2 electrons to it. We're going to add a hydrogen to it, and we get NADPH. Okay, so that represents a harvesting of energy. So what about ATP? We said we're going to generate ATP. Well, remember, we're building up a lot of hydrogen ions in the thylakoid space. So the hydrogen ion concentration is higher in the thylakoid space compared to the concentration of hydrogen ion in the stroma, which is a clear fluid that's all around. If you look at this picture, right, it's clear fluid all around these stacks of thylakoids. So what's going to happen? Well, we're going to use that electrochemical gradient. Remember the book we also had, it was also called the proton motive force. It's the same thing. These protons, because that's what a hydrogen ion is, the proton moves down its electrochemical gradient. There's actually a spinning thing that we saw the video of this. It spins around and around. ADP and inorganic phosphate enter in, and we get ATP out. Okay, and this whole process here of taking this electrochemical gradient and using it to make ATP is called chemiosmosis. Okay, so it's a specific term for this thing here. So we saw a lot of these elements before, right? When we talked about aerobic cellular respiration, right? Okay. All right. So, in fact, here's a picture. I'm not going to go through it, but there's a lot of similarities. Okay. So that's where we ended before. Oops. So now we're on to the Calvin cycle. So humans and most other organisms release CO2 during cellular respiration. So there's CO2 out there in the environment, in the air. The Calvin cycle are also called the light independent reactions or dark reactions. Is a series of reactions that use CO2 from the atmosphere to produce carbohydrate, which is going to be sugar, such as glucose. The carbon cycle reactions occur in 3 stages. So these are 3 important stages. The first part is carbon dioxide fixation. So what does the term fixation mean? Well, you have carbon and carbon dioxide, but it's not really very useful to us. In fact, it's a waste for us, right? Fixation means you take an inorganic molecule like carbon dioxide and you turn it into an organic molecule, or you attach it to an organic molecule, and then it suddenly becomes much more useful for life. Does that make sense? We have a similar term for nitrogen. There's plenty of nitrogen that you're breathing right now. Most of the atmosphere is nitrogen, right? Roughly what we're breathing right now is roughly 21% oxygen, 78% nitrogen, and 1% inorganic, not inorganic, inert things like argon and so forth, right? So we actually have a ton of nitrogen, but it's not really available to us for useful things because it's not fixed into an organic molecule, okay? So the first step is we have to do fixation where we attach this carbon to an organic molecule. So we'll talk about that. It's catalyzed by an enzyme called rubisco. Rubisco is a kind of a slow enzyme. It's found in the plants, the leaves of all these plants. And you can imagine if you look across the earth, there's a lot of leaves. It's a slow enzyme, so plant leaves have a tremendous amount of this enzyme. Most enzymes work very, very quickly. This one happens to be quite slow. And so because of this reason, because of all the leaves on the planet Earth, rubisco is actually the most abundant protein on Earth, if you do the math and calculate it. All right, so then the next step is carbon dioxide reduction. So we're going to add electrons to this carbon. At the same time, hydrogen will go with the electrons. And basically what we're going to be doing is making a carbohydrate. So this will then be used, this carbohydrate will be then used to make sugar. And finally, we need to go back and make this chemical RUBP, ribulose 1,5 biphosphate. So this is an organic molecule. It's a 5-carbon organic molecule. In the 1 and 5 positions, that's what this 1,5 indicates. There are phosphate groups. So it's ribulose 1,5 bisphosphate. We can just call it RUBP if we want. Okay. So we're going to regenerate that molecule because this, I didn't mention it earlier, but this carbon dioxide is going to be attached to this organic molecule, RUBP, which again is a 5-carbon organic molecule with 2 phosphates. And then what we're going to get out with for each cycle of the Kelvin cycle, we're going to get a little bit of product, which we're going to use to make sugars. Okay, so we'll go through it now. Okay. Before we get into the diagram, let's just quickly go through this. The reduction of carbon, CO2 to carbohydrate, CH2O employs light independent Kelvin cycle or dark reactions. These reactions are also known as C3 photosynthesis. See, because the CO2 first combines with a 3-carbon molecule. Oh, okay. I thought it was 5-carbon. 3-carbon molecule, ribulose 1,5 biphosphate in this cycle. Only 1 carbon is added per turn of the cycle from the CO2, so the cycle requires 6 turns to make a 6-carbon sugar. Actually, this will be very clear. Like, this is an important point. So you have 1 carbon here in CO2, right? There's 6 carbons in glucose. So it makes sense you'd have to go through the cycle 6 times, right? You keep adding to make 1 glucose, right? Because 1 times 6 is 6, right? The Kelvin cycle also recycles the reactants needed in the reaction. So we're going to ATP will become ADP in an organic phosphate. NADPH will become NADP plus hydrogen. NADP, right. The hydrogen will actually get used up when we make the carbohydrate. It returns NADP, so it's available for chemiosmosis in photosystem 2, right? Yeah, photosystem 2, we just talked about it, right? It has the ATP synthase. It returns NADP back to the electron transport system of photosystem 1. So remember we talked about how NADP is an electron shuttle. It's getting electrons from photosystem 1, right? Okay. So let's look at this Kelvin cycle. So we have, we're going to start here with this thing here, which is ribulose 1,5 by bisphosphate. So we have 3 of these. Actually, I was right. It's a 5-carbon. Okay, that previous slide was wrong. I was right. So it's a 5-carbon molecule, right? So it's a 5-carbon organic molecule. I mean, it would make sense. It should have 5 because it has the phosphates attached to the 1 and 5 position, right? That's what the 1 and 5 means. So I'm not quite sure. Maybe I read this wrong. Anyway, let's go ahead with this. We have the CO2 coming in, and So really this represents, this is really in a sense represents 3 cycles, okay? Because we got 3 CO2 coming in. So this is, so we have 3 of these ribulose 1,5-biphosphate. To those 3, we add 3 CO2s. Now each of these is a 5-carbon molecule, right? Organic molecule. We add 3 carbon dioxide. Each one has 1. So 5 plus 1 is 6, right? So now we have 3 6-carbon intermediates. Okay, we care about the name of the intermediate, right? So this is the first step in which we have essentially CO2 fixation, because we're attaching the CO2, which is an inorganic molecule, to this organic molecule. So it's not shown here, but it has hydrogen and carbon and so forth. Okay, so that's the very first step. Now the next thing we want to do is we want to reduce. So we end up here. There's a lot of enzymatic reactions that aren't shown here, but we have some enzymatic changes here. We have now 6,3-PG, which is 6 of these molecules, 3-phosphoglycerate. And these are, what happens is this becomes split. The 6 carbon molecules become split. So instead of 3 6-carbon molecules, we have 6 3-carbon molecules, okay? So these are now 3P, the name of the molecule is 3-phosphoglycerate. ATP comes in, it's going to donate a phosphate group, so it's going to cause the change, and now we're going to have 6 BPG, and these are so 6 3-carbon molecules. So you can see actually that the name change makes sense. Here the phosphate was at the 3 position. Now we've added a phosphate. So we have 2 phosphate groups, so that's 1, 3. This is just indicating the positions, right? We have carbons 1 through 3. So and the bisphosphate is like a bicycle, has 2 wheels, right? So we got 2 phosphates added to this glycerate, okay? So now we're not quite there, but the next step is we're going to reduce this thing by NADPH, which is generated by the light reactions. These come in. They're going to donate the 2 electrons plus the hydrogen, and we go from this molecule, the BPG, to G3P, which is glyceraldehyde 3-phosphate. And this is actually the molecule. This is the sort of molecule we really want. This one here. This glyceraldehyde 3-phosphate, we've got 6 of them, because that's the one that we can use to make things like glucose. And from glucose, we can make fructose. We can combine a glucose and a fructose, and we have a disaccharide called sucrose and so forth, right? And we can build up lots and lots of other organic molecules. So that's kind of the output that we want. And so we're going to take one of these molecules. So from one here, we put in 3 carbon dioxide. So we're going to get out 1 3-carbon molecule. Does that make sense? We put in 3 C carbons here. If we subtract the carbons, we put in 3 carbons here. Out at this end, think of this as like a machine, and out of this end, we get 3 carbons, right? So we're counting for all the carbons. But that means we had 6 of these 3-carbon molecules. That means there's 5 left over, right? So we've got 5 G3Ps, and there's 3-carbon molecules. We're going to use some more energy, because what we're going to do is we're going to need to regenerate this 5-carbon molecule, the ribulose 1,5-biphosphate. Okay, so that's going to take some energy, and then we're eventually going to be using these to combine with CO2 all over again and do the carbon fixation, the CO2 fixation. So. Okay, so again, there's a reminder here that the ATP, where does it come from? It comes from the light reactions. The NADPH, it comes from the light reactions. Okay, so to make 1 glucose molecule, how many carbons are there in glucose? Does anyone remember? 6, right? So we're essentially going to have to do this cycle. 1 cycle is actually 1 CO2 going in. Here we're showing 3 cycles. So in total, we're going to have to do 6 cycles, right? 6 CO2s will have to come in to make 1 glucose. They're showing you 3 cycles altogether, just because it's much more convenient to do it this way, right? So you have to double what's going on. You essentially you add not 3, but 6 CO2s to make 1 glucose, okay? Again, that makes sense, like the numbers add up, right? 6 CO2s, that's 6 carbons. You get out 1 molecule of glucose in the very end, right? Okay. All right. Oh, yeah, and then here, I forgot to mention this, there's a RUBP carboxylase. It's also known as Rubisco. It's the enzyme that catalyzes the first step in this CO2 fixation, okay? So it's helping this catalytic reaction here between the ribulose 1,5 biphosphate and the CO2, okay? And it's a strangely slow. Most enzymes can work at the rate of like 1 million reactions per second. There's some crazy, maybe not 1 million, but a very high number of rate. This enzyme is quite slow, and that's why your plant leaves have so much of it. And it happens to be the most common protein that appears on the whole world in the entire Earth. Okay, so here we go. So this is just showing the cooperation of the light and dark reactions in photosynthesis. So again, we talked about this earlier. We have water being split. We're getting oxygen out, sunlight coming in. We're generating ATP and NADPH. These are going through the Kelvin cycle, which is the synthesis reaction. And then we're bringing in CO2 that's being reduced, and we're getting out carbohydrate sugar. And we're regenerating. So the NADPH becomes NADP, which is good because we need that, right? Otherwise, the light reactions would stop. And then we also need the NADP and the inorganic phosphate, again, for the light reactions to continue to work. All right, so that's kind of it. I think is there a movie here? Yeah, here's the movie. So we've gone through it step by step. Let's take a look at the movie.
When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose.
Okay, so that made some, at the very end, there was a couple of points that are really important. One is that we need 6, well, the whole thing was important. Like, you should revisit this video. It's actually very good. But to make 1 glucose, right, we need to do 6 cycles of the Calvin cycle, right? And that makes sense, because if you consider each cycle as the entry of 1 CO2, then you would have to do that 6 times to make a 6-carbon organic molecule like glucose. The other point that they made was this generation of sucrose. Sucrose, you guys know, you know what sucrose is, right? It's just your basic table sugar, right? That people put in their tea and coffee. You get it in sugar cane, sugar beets, blah, 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 right? All these different things. Maple syrup has a lot of sucrose. So that's a very important sugar because it's the one that plants use to move these sugars around. They don't really in the arteries and veins within the leaves of the plant and so forth, and within the trunk of the tree. They don't move glucose around or fructose around usually. They're usually converted into sucrose, which is a disaccharide. So glucose and fructose are very similar. So basically just they have to do some minor changes to make glucose into fructose. You combine glucose with fructose, and now you have sucrose. The other point they made at the very end there was, which is important for us, is because we eat vegetables, right? And they're delicious, it's starch, right? So starch is in fact, it's a polymer, right? We talked about this before. It's a polymer of glucose, right? So you find starch in lots of things, right? Like you find starch in pasta, you find it in corn, there's cornstarch, there's potato starch, and so forth, right? So that represents a tremendous amount of energy, right, for us as animals that eat plants. Cellulose, where do you find cellulose in a plant? The cell wall, right? So if you're wearing a cotton shirt, that is in fact pretty much 100% cellulose. Paper is cellulose, right? So there's different forms of cellulose, that we use for cellulose, but it's essentially just a glucose polymer, but it's an insoluble one, meaning it doesn't dissolve well in water, and it's very difficult. Like humans, can we digest cellulose? No, we can't, right? So it's essentially what contributes to what's called dietary fiber, things you can't digest. Okay. All right. So let's move. So we're basically at the end of this chapter. What I'm going to do is you can take a look at this. What I have is prepared for today is a sheet that we'll do together. And then I do have some questions using Compound that we can go through. Okay. So I'll pass out the sheet. So this is an in-class activity, so you should be able to, we'll go through it. I want you to all work on it, and of course you're very welcome to chat and talk and work it out with your neighbors. That's going to encourage it. And then we'll also go over it together and make sure everyone's got this ready. Remember to put your name on it. Oh, and we do have I think you guys saw, I sent out an announcement, so we do have an exam next week, right? So this Thursday, I'll be doing a review session within class time. And then we do have the exam essentially a week from today, right? On Tuesday next week. So as usual, you can look at these review questions and answer it. So people that are successful in this class often will take this seriously, go through this and answer all these questions, because these are, it's information that may end up as a short answer question, that sort of thing. Okay. You doing okay on this side? So remember, I need you to fill this out because I'm gonna collect these at the end of the. It's an in-class activity, so I need you to do it in class. And you can, again, talk to her if you're having, getting stuck, I can come and help you, but you can also, and you can also talk to people around you. What you know, so forth. You done already? You want to look at something here? This one? Yeah, so it reduces carbon CO2, right? That's the whole point. Like, that's the, we have the three steps, right? The first is fixation, right? CO2 fixation, the carbon fixation. Then the second is reduction, which means you add electrons and actually add some hydrogen to it too. And that's the point where you get the hydrocarbon, right? That we're going to use to make the body uses to do it, so the plant uses to make glucose. Right? So that's correct. Yeah. Well, hold on, let's do it. So, yeah, it requires chemical energy. Yes, because it's a synthesis reaction, right? And this is wrong, yeah. I think we got it. Yeah, that's correct. It produces carbohydrates. Right? The carbon cycle, that's the whole point. We're going to get glucose out eventually. Well, carbohydrate makes that three carbon carbohydrate. It reduces CO2. So remember, you have the carbon fixation of the first step, and then the second step is the reduction, and the third step is the regeneration. The 5 carbon. Again, well, I think that's correct. Yeah, that's right. You did the same thing. Yeah. What'd you do? I think everyone got
Oh, wait, yours was the session before. I'm pretty funny.
Well, just a second. So, this one, the inputs are NADPH and NADPH, so that's incorrect. These are correct. And then this is correct.
So that was fine.
What? Yeah, so just go ahead and circle. So this one not, but these three, right?
Yeah.
So let's start on this one. Okay. That's correct. You got those three correct. You're missing one. So it's this one here. If the current look at the dating one, then it's right independent. Everyone does that. So that was the one. The next one. Oh, yeah, that's right. That's right. Yes. Yeah, so you circle this one? No, and North temperature is in the right option. Between photosystem 1 and 2. And other than that, you got it. This one was from it's all right. And also because it's light independent, it can bring it to the daytime. That's good. You have the same thing.
Yeah.
Yeah. You had, okay, good. That was the one that's trouble. The other ones you're probably okay with, they're just basically copying from the picture. Did you get this? I thought that all of them. The other ones you got there, right? I mean, it's just basically copying right from the picture. Yeah, looks good. All right. I think one of them got it. We're still working on the diagram. One thing you can do is if you find it, so I have the PowerPoints uploaded to the Blackboard, you can find it through the PowerPoint. You can also find it through the, yeah, so this is chapter 7, right? You want to go to chapter 8. When we saw the regulations after, one of the after. Yeah, okay. All right, I think it looked pretty good. I don't, does anyone have a question? So for the first diagram, this and the last one, basically just look at the diagram, right? And just copy over, right? So it's really not hard. For the next one, circle 4 phrases that are related to the Kelvin cycle. It looked pretty good. You should have 3 that are circled all in a row. Reduce the carbon dioxide gas. Remember, the first step was the carbon fixation. The second step is the reduction of the carbon dioxide gas. Once it's been attached to the organic molecules, so the fixation, the reduction, and then finally the regeneration of the ribulose 1,5 bisphosphate. Produces carbohydrates, right? Because remember, we make a 3-carbon organic molecule. We combine 2 of those and we make glucose. It's going to require chemical energy. We saw that in the form of ATP. And you can also consider the NADPH, which has the 2 electrons. That has chemical energy too, right? That's a a high energy molecule. It donates the electrons, which again contributes to the reduction of the CO, the carbon and CO2 gas. And then finally, the last one can occur in the daytime or nighttime because it's light independent. Okay, so let's go ahead and we have a little bit of time. Let's do some questions. We're going to do a sort of a lightning round of some questions. So function of light reactions is to what's that? Oh, it's called top hat. And then there's a join code. There's 307988. And you don't have to do it. It's not for credit or anything. It's all anonymous. So I don't. But if you happen to have a cell phone or a laptop, you can do it. Otherwise, you can just sort of mentally do it, right? Insert, or you're also allowed to talk to people and see what your neighbors are answering. The function of light reactions is to do what? Obtain CO2, make carbohydrate, convert light energy into usable form of chemical energy. Degenerate ribulose 1,5 biphosphate. Okay. So again, I'm going, there's a lot of questions. That's why I'm going a little fast. Don't worry if you're not. Oh, okay. That's correct. All right, let's go to the next one. Okay, I'm gonna go ahead. Oh, everyone's answered. Very good. Okay, so in this case, for this question, this is all of them are correct, right? So the Calvin cycle produces carbohydrate, right? A 3-carbon organic molecule, which is used to make glucose. It converts one form of chemical energy into a different form. Yeah, so it does some conversions, right? It takes ATP, NADPH, and then you're going to make this carbohydrate. So there's a conversion there. We have to regenerate each cycle. We have to end up regenerating that 5-carbon molecule, which combines with CO2. That's the ribulose 1,5 bisphosphate. Use products of light reactions. Uses ATP and NADPH. So all these are correct. Okay, look here. You know, people answered, so it's kind of all over the place. The final electron acceptor. The final acceptor of electrons during the light reactions of the non-cyclic electric pathway is the NADP, right? It accepts 2 electrons. We have the enzyme NADP reductase, remember? And that's associated with which photosystem? Photosystem 1, right? All right. Let's go to the next one. So I posted this PowerPoint, by the way, right? So this is on Blackboard. So you don't have to, all these questions are given to you on Blackboard. By just downloading the PowerPoint. They're at the very end. I just put them at the very end after all those review questions. They're there. Okay, let's take a look. Again, there's a lot of questions to go. I'm not trying to rush you too much, but I just want to get through a bunch of questions. So, okay, again, people are sort of all over the place on that one. So the oxygen given off by photosynthesis comes from water, remember? So the roots bring in the water, it goes up to the leaves. And then we have the hydrolysis where the water is split. And then we get the oxygen. Let's go to the next one. So I'm gonna go a little faster on these last ones here, so I just want to get through a bunch here. I've got like 12 questions here, so. The ATP and NADPH from the light reactions are used to do what? Okay. Yeah, so it's the last one here. We're going to convert 3PG to G3P. So 3-phosphoglycerate, glycerate, I think, to glyceraldehyde 3-phosphate. Okay. Yeah, let's take a look. Yeah, so cellular respiration and photosynthesis do have some similarities. They both contain an electron transport chain, and they have hydrogen ion pumps, and they both use chemiosmosis. Okay. Okay, so reduction of oxygen does not occur. Right? So reduction of oxygen means you're adding electrons to oxygen, right? So where does that occur? That occurs with aerobic respiration, right? Remember, oxygen is going to find an electron acceptor. An atom or molecule that accepts an electron is being reduced, right? So that does not happen in photosynthesis. Whoops, what happened there? Did I not? Maybe I didn't put a. What's the correct answer here? Starts with carbon fixation, right? I think maybe I didn't put the correct answer in here. It didn't. Yeah. You know what? I put the wrong thing in here. That was me just clicking on the wrong thing. Sorry about that. So this is correct. Carbon fixation, people are correct there. Reduction happens second. Okay. So the second stage of Calvin cycle needs input of energy to reduce the 3Pg molecules that result from the first stage. Where does this energy input come from? ATP and NADPH that were supplied through the light reaction. Okay, so there's probably a few more, but you can see them in the PowerPoint that I uploaded to Blackboard. Okay, all right, thanks, guys. And I'll see some of you in lab later today, right? So remember to do your pool lab. Oh yeah, I'll take that from you. Maybe I'll just stand over by the door. I'll get your thing. Well, because they can both be nature of the. Yeah, I should be on as thank you for finding them. No, it's fine. How many times have you something about
Thank you.
You're supposed to understand that the teachers are, keep alive and okay. Yes, it's a side down to do, right?
Yeah.
It's a national mistake. You don't want to do that. All right, so are you making any progress? If I gotta send you the actual instructions, but have you found any more sources of information or anything?
No, I've been on like my paper for English and
reading.
All right.
Or general stuff.
Just keep it in your mind, we gotta start working on this. And I'm saying that to remind myself too, right? It's not just like,